In this video, we will discuss about the cGAS sting pathway can be used in p53 mutant cancer, blood cancers. So, the role of p53 is that whenever whenever any cells acquire characteristics of uncontrolled growth rate, then p53 first try to controls that uncontrolled growth rate. If it is beyond their threshold limit, then p53 will trigger apoptosis. So in most of the blood cancers, the p53 has found to be mutant. So because of that, what happens is that p53 will not be able to play a role to trigger apoptosis, and these blood cancers or mutant blood cells will continues to replicate. So what the researchers has find out, the role of p53 is that the p53 is used to upregulate these BH3 only proteins, and BH3 proteins belongs to BH3 family, and the role of this BH3 proteins is that it will suppress the pro-survival proteins, and these pro-survival proteins, since they are suppressed, they will not be able to suppress this pro-apoptotic proteins. So it will cause indirectly the upregulation of these pro-apoptotic proteins, and it will trigger apoptosis. This is how p53 normally works. But in the case of this mutant p53, what researchers has tried to do is researchers tries to mimic these BH3 proteins, or what we say they do the BH3 mimetics to inhibit BC2 family pro-survival proteins. So what they did is they created an environment where the cells will experience the upregulation of these BH3 proteins, and since the cells experience the upregulation of BH3 proteins, the pro-survival proteins goes down, and it will cause this pro-apoptotic proteins to go up, and then it will trigger apoptosis. This is how the researchers are currently working on. This is how the blood cancer cells are currently being diagnosed. However, the problem in this approach is that the apoptosis that has been observed in the blood cancer cells is very low compared to the case where p53 is already been p53 is not in the mutant form. So what they have observed is that since the p53 is not in the mutant form, the apoptosis that are triggered in the blood cancer cells will somehow further accelerate this p53 to produce more BH3 proteins, or we can say that there is a cascade effect that usually goes on where p53 is not mutant. And thereafter, all the researchers has find out the alternative to the p53 is this sting agonist, or this cGAS sting pathway. So this sting pathways, if this sting pathways gets activated, its role is or its function is to further upregulate these BH3 only proteins. So the BH3 only proteins are upregulated, and it will do the same role that is done by this p53. That is, it will suppress the pro-survival proteins, and the pro-apoptotic proteins will go up, and it will trigger apoptosis. So in this case, the third part, that is sting agonist, can be used to induce BH3 only proteins expression in a p53 independent manner, thereby boosting apoptosis signals. So these sting pathways will plays a role of p53 in the p53 mutant blood cancers. So this is what the researchers has presented in this paper, that the role of p53 can be taken care by the sting agonist. But this and we will discuss in this paper how this sting agonist can be activated, and what are the results that researchers has observed to come to this point that this cGAS sting pathways is a alternative to a p53 mutant blood cancers towards upregulating these BH3 only proteins, so that we don't have to artificially mimic these BH3 upregulations in the blood cancer cells of patients who are affected with the blood cancer. So I hope you understood this graphical abstract that is presented in the paper, and if you need to understand more about this papers or abstracts in detail, you can go through the reference that has been provided in the description. that's all for this video. Thank you for your time.